Welcome everybody to BioPhorum's Cell and Gene Therapy webinar focusing on CGT process maps. I'm Stephen Wall, one of BioPhorum's facilitators, and I'll be hosting this event, although you will hear mostly from Jürgen Magnus of Bayer, and who will be taking you through the process maps work. But before we get into the main activity of discussing the process maps, I'd like to briefly introduce those of you who may not be familiar with BioPhorum. So at BioPhorum, our mission is to create an environment where the global biopharmaceutical industry can collaborate and accelerate the rate of progress for the benefit of all. And BioPhorum does this by bringing experts like all of yourselves together on calls like this, where you can identify the problems you want to discuss within your industry, and you can work together collaboratively to solve these common problems and generate whatever outputs, deliverables you would like to achieve. So this could be discussions, it could be white papers, position papers, or members-only surveys. So we hope by enabling this environment where you can collaborate, it will accelerate all of the activity to benefit. We currently, in BioPhorum, have 9 forums or topic areas, which you can see on this slide. These are the high-level forum groups. And we have, starting here, hopefully you can see on the screen, the development group. We have a group focusing on drug substance aspects, fill finish, information technology, medtech, a supply partner group, our newest forum focusing on sustainability, technology road mapping, 5-10 year forward thinking, planning and strategy for that, and cell and gene therapy forum, which is what we'll be focusing on today. Now, the majority of these other forums outside of CGT focus on general biologics, MABs, small proteins, with the odd exception where they do a small amount of work on CGT. However, within our cell and gene therapy forum, that is where we focus on all aspects that are associated to CGT. So a little bit about the CGT group. As I just mentioned, we do cover all aspects like the other forums, from early development, commercialization, supply chain, manufacture, regulatory, all the way to fill finish and beyond. And we have broken up into these 7 work streams within the CGT forum. We have the commercialization, CGT manufacture, one of the outputs being the process maps you'll be hearing in a little while. Environmental health and safety and biosafety, high-level analytics, raw materials, regulatory, validation, and our most recent focus topic, RNA, working on mRNA therapeutics and as a tool. And as you can see, within these 7 areas, we are covering the full range of CGT challenges and problems to work together. And in the RNA, we're doing that again, but focusing on the unique RNA problems. Now, as with all the other forums across BioPhorum, within CGT, we work together, we have lots of cross workstream and, just as importantly, cross-form interactions. So if we can leverage, utilize work that's been performed by other forums, other teams within BioPhorum, or we can work together and to make sure that the best people are together working and leveraging the best content, so we could just add value content, not duplicating anything. Now, as you can see on this slide, the CGT forum started in 2018, and in its almost 4 years, we have grown from 16 companies to 43 member companies, and we now have over 700 participants across those member companies. During that time, we created a wide range of outputs, 21 external publications and presentations at meetings, and 2 of which you'll be hearing in any moment from Jürgen, the process maps. Now, all of these topics, and including the CGT process maps, have a great need for collaborative discussion and standardization that will only help the industry make optimizing processes and quality aspects easier and better. And so this is what BioPhorum is trying to help all of our members and the industry as a whole to achieve. As I said, today's webinar will be presented by Jürgen Magnus of Bayer, and he'll show examples of some of the CGT process maps that the previous CGT mapping group have created and published. and how these can be used to help design of company-specific processes and production facilities. Now, before we get into it, I'd like to highlight there will be set points during the talk where Jürgen will pause and there'll be opportunities for Q&A there. But I will also be monitoring the chat. So if you'd prefer to put your question into the chat, please do so. And then at those pause points, I can voice them on your behalf, or you can mention them. And also, if you see a question in the chat that you think is really good and you'd like to vote for it, please add a thumbs up to it. And then if we have lots of questions, we can prioritize those with lots of thumbs up. So before we get into it, I would first of all like to introduce Jürgen. So Jürgen holds a master's in chemical engineering and a PhD in biochemical engineering. He's worked at NNE Pharmaplan with the design and construction of pharmaceutical production facilities. And since 2009, he's been with Bayer in roles linked with production, technology, and facilities. Following his 5 years in the bioprocessing technology group head role, he is now part of BioPhorum of Bayer's CGT unit as a strategic manager. As you can see on this slide, he has a wide range of interests associated with CGT, some of them shown here, and he brings his great experience, interest and energy to BioPhorum's collaboration, and he's been a member since 2013, including this CGT process maps activity. And he's been a key contributor and driver to this. And so I'd just like to thank him for all of his content, agreeing to give today's webinar, and just really thank you again, Jürgen, and please feel free to take the screen and take it through the next set of slides. Thank you.
Yes, thank you very much, Stephen, and I hope that you can see my screen. So
yes, thank you.
Take you out there as well. So yeah, thanks for the introduction. And before I start talking about the gene therapy maps and the cell therapy maps, I just wanted to say a few words about the motivation for creating them. And I think that we know that the cell and gene therapy space is moving very rapidly. There's a lot of therapies, hundreds of programs in clinical development, but not so many in commercial production. And so this wave is now coming into the commercial production. But it seems that we didn't have time to develop production technology that is suitable for large-scale production, and at least that's my opinion. So as it says on the right-hand side here, the lab results were so good that we bypassed process development, and that is very much the feeling that I have when I look at what we're trying to do in the large-scale manufacturing. So there has been some new facilities, large facilities being announced recently, and it seems that the industry is ramping up production capacity really at the large scale. But we still need to do a lot of work in developing processes that are suitable for large-scale commercial production. So in that respect, it feels a little bit, if we compare it with the antibodies and how that developed, if we remember that in the beginning of the antibody development in the 80s, people used a very different technology than what they do today, and they used to do roller bottles, and then when they wanted to produce in large scale, they just used more roller bottles. But then this developed, and today people think of roller bottles as an ancient technology that no one wants to use anymore. And maybe cell and gene therapy is going through a similar development, and that in the way we are now in the 80s, so to say, of the antibody development, and then in the next 30 years we will see hopefully a very big change in how we process cell therapies and gene therapies. So just another slide to highlight this a little bit. If we look at adherent cell culture, then if you are in a research setting, you have 1 or 2 plates that you can use, but when you try to scale it up, then what you do, you just add more plates. So that's not really a scale up, it's a scale out. So you have here, in this case, 40 plates on top of each other. And then when you need more material, you combine them with another 4 stacks. So that gives you then 160 plates. And then you put that into an incubator with 3 other carts, which gives you then 4 times 160, which would be then 640 plates in total. And then you can supply a few hundred patients, but you're using exactly the same technology as you do in the research scale. You just have an enormous amount of work to move these plates in and out of the incubator into the biosafety cabinet. And you have very poor control of the process conditions in these plates, because you don't control pH, you don't control dissolved oxygen, even temperature will change, because in the incubator you have 37 degrees, but then when you take them out of the incubator, you're probably at 21 degrees. And then your cells are experiences variation in temperature, and maybe that is also different. Maybe the plates at the top and the bottom have different temperatures than the one in the middle, because they have a different surface to transfer heat, etc. And all this uncertainties in a very complex process. If this is an iPSC process where you have iPSC cells going through a differentiation process, you have to change the media many times during that process, and you have all these variations in the process parameters. This is clearly not a good way of producing it, and you don't have the quality control that you would like to have. So someone needs to come up with a better solution, and suspension culture could be one solution. There has been also some advances there, and in that case you can scale it up. So that's where you can make larger bioreactors, eventually going into a large-scale bioreactor like a 2,000-liter bioreactor. But it's not obvious that suspension cell culture is the answer either, because let's say if you develop a tissue type that is not naturally in suspension, like muscle cells or something, it's not clear that suspension culture is the right answer either. So I think that it feels a little bit like the roller bottles all over again, and that we are hopefully going to move into something that is better than this. And the process maps can help us as an industry to get consensus and to have a common reference frame, which is really the reason why we created them. And we thought that industry can work together on improving processes in the pre-competitive space, because it's not necessarily linked to any therapeutic programs. And so from having the state of today, where it doesn't seem to be really a common standard for industrial production, through the BioPhorum, you bring the people together that can collaborate on this and create then this common reference frame, which can be then very helpful in developing the next generations of processing technology. So that's why this was meaningful to create these process maps. And let's start by looking at the gene therapy process maps. So we had here a good group of people from some large companies like AstraZeneca, some of the front runners, I would say, in gene therapy, like REGENXBIO and Sangamo. And also importantly, we have had Lonza participating in this as well, which is a CDMO. So we had the view of a toll manufacturer as well, and got a good group together to work on representative process maps that we feel represent the most important or the most common types of gene therapy processes. And you can download it, by the way, or everyone can download it from the BioPhorum internet page. So how did we approach this? This is a busy slide, but it's an overview of the different types of AAV processes. So on the left-hand side, you can see the process steps or the process stages. And then we have here the different types of processes or different expression systems, if you like. So first we have the HeLa system with the adenovirus infection. Then we have the SF9 baculovirus system with a co-infection, where you can also use herpes simplex virus with the Vero cell line. And then the most common expression system is the HEC293 triple transfection process. And then we also included a process for the production of lentivirus, which is used then mostly within cell therapies. But these three or these four different processes here, they are different and therefore have different processing steps. In the HeLa case, for instance, you have the RepNCap genes as well as the vector genome on the chromosome of the HeLa cell, and then you add the adenovirus to start the production. But that means that virus safety and virus removal steps are important in the processing. So the whole process of downstream processing, but also how you prepare the cell banks and also how you prepare the transfection, or in that case the infection solution, is different. In the baculovirus case, you also have two different types of virus that you also need to remove before you don't want them in the final product. Whereas for the HEC293 case, it's really the plasmid, so the virus is less of a concern, but then you have to focus more on removing this plasmid that you're using in the production process. So each of these processes will have different processing steps, and in the document from the BioPhorum, you can click on these boxes, and it will then take you to a more detailed process map of each of these different steps here, depending on what your process is. So one example here is the cell bank and plasmid preparation for a transient transfection, where you see the different steps of the working cell bank for the HEC293 cell line, but also even the production of plasmid if you want to produce that themselves, and the purification. And then the process map is then linked to the next step by these yellow cones here that will then take you to the next processing step. So that's one example of this. Here is another example of downstream purification, which of course then is also linked with the upstream part and then continues to the filling and the drug product part. But here you can see the different processing step in that sequence. And yeah, so the process maps consist of these type of block diagrams. In addition, there is some information about the function of each processing steps, and also the equipment that you typically use for these processing steps. So that's something that you can also read out of these processing maps. And moving on to the cell therapy, so there are two different documents. So one is for gene therapy and the other one is for cell therapy. And here we had many of the same companies, but different people, interestingly. So also a very good group that contributed to this and hopefully gave a good picture of what industry thinks are the relevant processes. But in the case of cell therapy, we distinguish first of all between autologous and allogeneic cell therapies, and this is of course a very high level first introduction to this, where in the autologous case you collect the cells, you manufacture the product, and then you administer the cell to the patient. In the allogeneic case it's a little bit different because you have donors, and it's less time critical because it's not patient specific. But this was the first distinction that was useful to make between these two types of cell therapies. And what we tried to do when we created process maps was to also distinguish between, or to include both processes that do genetic modifications and those that don't do that. So I think the next example is an example of an autologous cell therapy where you have gene editing using a virus. And so here you have the case where the patient has donated cells maybe somewhere else than where the production facility is, so that the cells had to be frozen and then shipped to the production facility. Or in the case that that you have something like a local production directly in the hospital or something, it will be then the fresh cells that hasn't been frozen. But in any case, these are the different processing steps that they go through. You have the viral delivery, typically a lentivirus used for that step, and then after the processing, you ship them to the administration site. So that's one example of autologous cell therapy. And here's another example of an iPSC differentiation process, where filling is also part of that process, and and it's the same principle here, that you go through the different. This of course assumes that you have generated your cells before, and that happens then in the previous process maps, but then the differentiation process is really shown here, including also the filling of the cells in vials. And also here there are a lot of information relating to each of these processing steps as well. So this is how the gene therapy and the cell therapy process maps are built up. And I don't know, Stephen, if this was the point where we wanted to ask for questions. If not, I can continue.
No, I think, thank you, Jürgen. Slide echo there.
Okay, I'll continue then.
Unless there's any questions to everybody. I know everyone's on mute as well, Jürgen, but if anybody would like to just put their hand up or indicate in the question if they have any specific questions to the maps before Jürgen moves on. No? Okay, please add them later if you'd like. So Jürgen, thank you so far. Keep moving on.
Yeah, thank you very much. I just wanted to show an example of how we actually use this in my company in Bayer. We designed a production facility for production of viral vectors, AAV vectors for gene therapy. And then we started with this high-level overview that I showed on the previous slide, which, because in our case, we didn't want, we wanted a multi-product facility where we didn't want to specify it for one particular process. So we had to be able to process different types of AAV expression systems. So that's what was kind of the starting point. And looking at the more at the block flow diagrams that process maps provides, and then we went into more company-specific design, where we really went into each and different unit operations and specified them, the size of the equipment and other things, and then based on that we could go into the design of the facility itself. So just to show, this is some work that we did together with Novo Nordisk Engineering. So it's a typical layout that they use, but here you have a more detailed picture of the process, and in addition to this, there's a detailed text, process description that explains this, and then this became the basis for the design of the core process, that we then designed a facility around that core process. So here is what came out of that exercise, where you have the bioreactors located here. These are 2,000-liter bioreactors. Then you have the downstream area here with the chromatography units and filtration units, etc. And then there's a small filling room here with a small isolator and a filling machine inside it. And here is the buffer preparation and the media preparation. So this design is sometimes called an outside-in design, because you have the clean corridor on the outside, and so the raw materials move in the clean corridor into the buffer preparation room and into the processing rooms, and then the waste, the used disposables, is collected in the center of the building here in this waste corridor, where it's brought into an autoclave and taken downstairs to be removed from the building. So that's the design that was chosen for this, and I think that we ended up with a very efficient design. We put the whole production just on one level, and that would be level 1, and then we had on the ground level the gowning and storage, and people who worked in these 2 different teams. So there was a clean team that deals with the raw materials and the product, and a dirty team that deals with the waste and the used disposables. So it became a very compact design, and the building itself and the facilities is quite small, considering that it has the capacity of 4 2,000-liter bioreactors. So that was the outcome of this, just as an example of actually how we used the approach of the process maps in this context as a starting point, really. So I am about to hand it back to you, Stephen, but maybe just to round it off a little bit with an outlook from my side. The process maps can be useful for specific cases, as I just tried to give an example of, but of course, hopefully this helps us create consensus in industry, and is helpful in the collaboration among the different industry partners in this development of a better production technology that I think is very important. And I think that as an outlook, that could continue on a more detailed level, and that we can then continue to converge towards the better processing technology, which is just a benefit for everyone.